All right, so today I'll present a little bit on how differential expression of these rhizostomin pigments lead to color variation in a jellyfish. So just a little bit of background. Color is a very conspicuous feature of many animals, and this can come either from structures, like here in the wings of these butterflies, or it can come from pigments, which are these chemical substances that selectively absorb light. And these pigments, they can either be produced by the organisms or ingested. As you can see here, this Fiona nudibranch is getting its pigments from feeding on the velellas. Sorry, Rebecca. And these pigments, they have very important functions for the survival and reproduction of this organism. And some of the most important ones that we all know are like camouflage, thermoregulation, and photoprotection. In jellyfish, the most common types of pigments are carotenoids, like the blue colors of velella and other hydrozoans and scyphozoans. The red-brown coloration of Periphylla, Anatola, and other deep-sea medusae come from these tetrapyrrole pigments. Melanins, like the magenta-brown, as you can see in the spots there in Pelagia, and the very famous green fluorescent protein family that was first discovered in Equoria, but that's distributed across hydrozoans. And everyone knows at this point that this was, there was a Nobel Prize in chemistry in 2008 given to the green fluorescent proteins. Not that I'm expecting a Nobel Prize for studying jellyfish pigments. But this was very significant because this completely changed biomedical research, because nowadays you can use these fluorescent proteins as tags for observing in vivo processes in cells and tissues. And so we saw a few talks on regeneration, and I think the talk by Tenkara yesterday as well, using these fluorescent pigments to be able to observe the bacteria and process in regeneration. And if you want to know more about the diversity of pigments in jellies, come see me at my poster outside today. We can I'd be happy to chat and hear more about your ideas. But beyond these common types of pigments, there was a new class of pigments that was discovered from Rhizostoma pumo in 2004. So this group, they isolated and purified and sequenced the blue pigment from the bell of Rhizostoma. And they found that this was a protein pigment that consisted of these two interestingly named protein domains, Kringle domain inserted within a frizzled domain. And this was never seen in pigments before, so it was a completely new class of pigments that was discovered. And previous to that, the only place that these two protein domains were found were in these ROR receptors. And so this is a schematic of a cell, and you can see those ROR receptors there on the cell membrane. And these ROR receptors, they're very important for these Wnt pathways. And so these are very conserved developmental pathways. And once that Wnt protein binds to that ROR receptor, there's a cascade of reactions that elicit these very conserved developmental processes. And in cnidarians, it has been reported that this is related to oral-aboral differentiation, similar to what the homeobox genes are doing in bilaterians. And so in an initial study, what we wanted to do is see the extent of distribution of these rhizostomin pigments in cnidarians. So we searched for these two protein domains in the genomes and transcriptomes of 93 cnidarians, and we found that that structure of the Kringle domain inserted within this frizzled domain was only found in rhizostomes. And so we established this new pigment family called, very conveniently, the rhizostomins, and we published this a couple of years ago. And so the very interesting thing about these rhizostomins, as you can see here, they not only occur in blue jellies, so also red and brown jellies possess them. And a really interesting thing that we also found is that some of these red and brown jellies, they possessed multiple rhizostomins. And including here Rhopilema, there's a congena, the Rhopilema azamuchi, that can have blue coloration. So one of the things we wanted to investigate further is there a functional diversification of these rhizostomins, right? So are these different rhizostomins possibly responsible for different colors? What are the mechanisms of regulation of these pigments? And if they could be related to intraspecific color variation. And this is important not only to understand the evolution and function of color in these animals, but also to look at potential applications of these pigments, because as many of you may know, blue pigments are relatively rare in nature. And so our target species for this study was the blubber jellyfish, Catastylus mosaicus. This is pretty common in Eastern Australia. And as you can see here, it can vary in color in two different ways. So the whole jellyfish can vary in color between this beautiful blue and a light brown or even a bit whitish sometimes color. And there's also this band around the bell margin, which is common among rhizostomes and also a little bit among the oral arm margins. And so we know that this coloration is not due to endosymbionts. So the first thing that we wanted to do in this study was to confirm if they had the rhizostomins that we expected. And so to do that, we initially started with a reference transcriptome. So we did that pulling tissues from different tissues from the different color morphs and also polyps and ephiri, and searching for these two protein domains in this reference transcriptome. And so as you can see here in this alignment, we found 4 rhizostomin candidates. So that means that these 4 sequences, they had that same structure of the kringle inserted within the frizzle domain. And it fell within the expected range of molecular weight for this molecule. So it's a pretty big protein, around 27 to 32 kilodaltons. So now to confirm if these rhizostomins were actually related to the pigments that we were seeing, we proceeded with protein characterization, which was the trickiest bit of the project. So for example, here with the bell margin pigment, I isolated, extracted these blue pigments and proceeded with size exclusion protocols such as HPLC, as you can see here, to purify that pigment. And this is the initial result from that. So you can see here, let's see if the pointer works, kind of. You can see here on the x-axis the different fractions that come out of the HPLC. And the bars correspond to absorbance along the blue range, so they're colored. You can see here that the bluest fraction that we obtained was also the one that we got this signature from mass spectrometry for this specific protein with an ion abundance. And we found that when we isolated this protein and went through mass spectrometry, we found that it had 28 kilodaltons, so within the range that we were expecting for the rhizostomins. And so we proceeded with protein sequencing, so using tandem mass spectrometry, and we were able to sequence 3 peptides from the rhizostomin, from the bell margin pigment, and we found that they matched exactly one of the rhizostomin candidates, which was a major relief for me. And we also found that the predicted protein sequence had 28 kilodaltons, so we were able to confirm that this sequence was the bell margin pigment. So now with the differential expression study side of things, so we'll start here with the differential expression of the rhizostomin candidates. So in this case, comparing tissues with and without that bell margin pigment, and as expected, the rhizostomin from the bell margin pigment was differentially expressed. So the bars here are showing that it's more highly expressed on the tissues that have the bell band compared to the tissues that don't have the bell band. So this confirmed our expectations for the bell margin pigments. And now comparing all of the tissues between blue and non-blue jellyfish. So we found 2 differentially expressed rhizostomins. One of them here on the left side was more highly expressed in the blue tissues, and also doing protein characterization, we were able to confirm that as the blue pigment, that rhizostomin, a 27 kilodalton protein that fell within the range that we expected. And we found that this one was more highly expressed in the non-blue tissues, but we were not able to confirm if it was the brown pigment or not. The brown pigment was a little bit trickier because it wasn't as concentrated in the jellies, so we had a harder time to try to isolate and purify the pigment. So unfortunately, we weren't able to confirm if this is the brown pigment or not. But we did find that these 3 rhizostomins were differentially expressed among the color variants. And so looking here at all the differentially expressed genes, and I'm focusing here on the blue versus non-blue jellies. So for those of you that are not used to these plots, I'll highlight here. On the top here are the top differentially expressed genes in the blue jellies, so the most highly differentially expressed genes in blue, and in the bottom here are the top differentially expressed genes in the non-blue jellies. And so the trend that we found among the highly differentially expressed genes between each of the 2 were a lot of extracellular matrix proteins, so a lot of the proteins of the mesoglia, as you can see highlighted here on the dots, and also a lot of proteins related to those signaling pathways, like the Wnt pathway that I mentioned in the beginning of the presentation. And so combining these two things, this pattern of the mesoglial proteins with these signaling pathways, points to this idea that there's some morphogenetic processes that are related to that color variation, stuff like body patterning and regeneration. And so our hypothesis here is that the signaling pathway was co-opted to regulate pigmentation in these jellies. But this is not necessarily related to development, because this can also happen during stress response. And so there's quite a few studies. One example here with heat stress in corals that shows that these Wnt and ECM genes were downregulated in these corals. So there could be perhaps some level of stress response that's related to color variation in these jellies. And so a summary of the presentation. We did find there was some level of functional diversification among the rhizostomins. So we did find different blue rhizostomins in different parts of the body, even though we weren't able to confirm if there is a brown rhizostomin in this species. Our hypothesis for the regulation is that the signaling pathways like the Wnt and BMP, very conserved pathways, were co-opted to regulate pigmentation, and that this is in fact related to intraspecific color variation. So 3 of the rhizostomins that we found were related to color variants in Catastylus. And so further research, we still don't really know what the function of these rhizostomins could be. So one of the hypotheses that's been out there, and it's one of our current hypotheses, is that this is related to photoprotection. And so it's been hypothesized for Cassiopeia. And maybe you've seen this, people that work with Cassiopeia, that sometimes when these jellies bleach, they become a lot more blue, not just because the blue is more evident. So this is something that we're investigating at the moment. And this is not unprecedented for cnidarians, right? So corals, expression of GFP in corals is also related to exposure to radiation. And also the applications of these rhizostomins. So there are a couple of groups in Mexico that are studying the potential application of the blue pigments of Stomalophus to be used as dyes in the food and beverage industry, because they're pretty pH stable pigments. And this could even be taken advantage because there's already a jellyfish fisheries established there. So this could be an extra source of income for that fisheries extracting those pigments that can be used. And the really cool thing is, because these pigments are proteins, that means that we don't necessarily have to harvest the jellies to get those pigments. So just like GFPs, the way that GFP are produced is that they're normally cloned by bacteria. So they're synthesized in the lab. So this being proteins makes it easier, potentially, for any applications, means that we don't have to disturb the jellies to use these pigments. And that's it. I'd like to thank all the amazing team in the labs that I've been very fortunate to work with. And thank you very much for listening.